Okay, I'd like to call to order the meeting of Amphipods Anonymous. I'm Jay, and I'm a recovering amphipodologist. This is always the most fun part of any advisor's job, is to be able to get to sit back and see in one place the awesome work that their students do. And so it's my great privilege to be able to introduce Claire Murphy to you today. Claire was one of the COVID kids. She interviewed here about 3 weeks before the world shut down. And fortunately, she couldn't interview anywhere else, so she came here. And she has been working, as you're going to see, on sort of connecting organismal traits, sort of animal behavior, and community ecology in ways that I think get at some really fundamental and what seem like obvious questions in ecology that turn out to be really hard. And ecologists, of course, will know that every ecology question is actually really hard, as it turns out. But what I think is most remarkable about Claire, and I promise I'll keep it brief because this is your show, not mine, is yeah, this is it though. Don't expect any more. She has this sort of really amazing way of blending really good, deep natural history of organisms that are kind of hard to study, as you're going to see, with sort of very cool field experiments, and then sort of forging her way through morass of statistics and sort of semi-creating her own way to do analyses that I think has led to some really cool insights that she's going to share with you today. So I am going to sorely miss her in my lab as someone who integrates all of those things and provides a good sort of reality check for me. Sometimes when I get too enthusiastic, Claire will settle me down a little bit. But I think you're going to see this come together really nicely in today's presentation. And then Claire will be off, before too long, off to a postdoc at UC Santa Barbara. And I think she may tell you a little bit about that. So not only do I get to announce my student leaving, but she's leaving to go to a job. So, you know, that's great. Anyway, Claire.
Yay, So today I'm going to talk to you about what I've done for the past year. So I think a lot about predation. As ecologists and evolutionary biologists, we also know predation is important in structuring communities. So we have pies here on the rocky shore eating mussels. And we have lions in the savannah eating all the things in the savannah. And not only are these sort of like static measures, right? Predation is really variable in different systems. So Eric Sanford found that under really small differences in temperature, you have different amounts of predation from pies on mussels, and it has these big impacts on the mussel community. And people have studied lions and seen that under different amounts of moonlight, so different parts of like the lunar cycle, the ability of lions to be able to see their prey changes, so the predation risk for different prey changes, and that can cause all these cascading effects throughout the prey. So I've been interested in sort of this big question of how might predation risk vary. And I work in seagrass systems, so we're going underwater. So this is seagrass. It's this really beautiful underwater flowering plant. It provides lots of cool structure, and there's lots of animals that live in it, which is what I'm most interested in. So if you think about a predator-prey interaction, the prey that we might think of is a little amphipod. So all of these drawings are by Colin Gross. So thank you to Colin Gross, because they're like the basis of my entire dissertation. And so if you Google amphipod, this is sort of what you'll find, this little like gamma ray guy. They hang out in the grass, they use it as a place to hide, and then they also forage and they'll eat things off the plates. And they're hiding from their predators. So if you think of a predator, a classic predator might be this rockfish. So seagrass are this nursery habitat for lots of species in the summer. Juvenile rockfish will come in and they'll forage through the grass. So rockfish eats the amphipod. But we have more than just one sort of amphipod living in the grass in this prey community. So you have these gamma ray amphipods. We also have these guys, these like stick bug sort of things, are coprolid amphipods, and then we have isopods. And we also have a lot of different kinds of fish, right? So you might have a big juvenile rockfish, but you might also have these fish with smaller mouths or crabs. So there's a lot of diversity in both of these communities. And then the habitat that they're foraging, that this interaction is happening in, also can change both across the year in temperate systems like in seagrass or Zostria, the seagrass that I've been studying, but also spatially. So it might not be quite as dense of a place for things to forage and hide. It could also be pretty open. So if we think about how predation risk might vary, we can think about it through sort of 3 different aspects of variation. So prey traits, habitat structure, and the predator community. And then all of these we might expect to vary also across the year and through space. So I tried to look at all of that sort of one at a time, and I'm going to tell you about each of these different pieces as my 3 chapters. So first, I'm going to talk about prey traits. Okay, so measuring predation is actually pretty hard, right? Like it's lots of things get eaten. It's pretty important in a lot of systems, but actually figuring out what is the strength of predation is difficult. And so we use these like predation assays. We'll use some sort of model to represent a whole prey community and see how often that's getting eaten. So in terrestrial systems, sometimes we'll use these clay caterpillar, right? You make a little model caterpillar, and this can represent predation on the whole insect community. We might use these artificial nests, so you can take chicken eggs, dye them different colors, put them in nests, see how often the nests are getting eaten. And in marine systems, we might use something that's like this. So this is a garden stake in some seagrass bed with, at the very top, you can see a tiny little piece of dried squid. It has a fun name, it's called a squid pop. And so we can use squid pops to sort of get a measure of predation risk over a really broad area or over a long period of time instead of having to figure out what are all the members of the prey. But if we think about these squid pops, and we think about what they're representing in the seagrass system, this little piece of dried squid looks very different and has many fewer traits than all of the different things that it's representing, right? So we have this very diverse prey community. And so I have most of my work is going to be about these 3 species, so you'll be very familiar with them. These in the seagrass apofauna community, we have lots of little bug things crawling around, but these are sort of the most common across all of the sites I worked at, and also the most common across the year. And then also they represent these dominant strategies that different members of this apofauna community use to avoid getting eaten. And we can think about them on this spectrum of the value that they're giving to a predator, so how much energy a predator might get if it eats it, versus the defenses that they have to avoid getting eaten, so how hard it would be to catch. And we put in these fun figures. So caprellas, we can think of them individually, they're sort of like low value. So you can see that, right? They're these little stick bugs. There's not a lot of energy sort of in each individual one. But they also don't have many defenses sort of on their own, right? It seems a lot of the time, it seems like they're at really high densities and they might use aggregation to avoid getting eaten. So if there's lots of them, then every individual one has a lower risk of getting eaten. The amphithoids, they are bigger, they have more biomass. I always just want to call them a jelly bean when I try to describe this. They're like a little juicy thing. And they have sort of medium levels of defenses. So they don't have a very thick shell. If a predator goes and tries to chomp it, it's pretty easy for it to bite it. But they do have silk glands on their back legs. So they can make these temporary tubes, and they can sort of hide and find algae or seagrass to make these tubes and hide in. And then the isopods are the biggest members of the community, the most biomass. If a predator could eat it, it's a lot of energy, but they also have the strongest defenses. So they have a very thick shell. They look a lot like the seagrass, and they do this thing where they'll find blades that are the same width as them to camouflage. So just if you're not familiar with them, this is the caprellids. So I'm going to talk a lot about them. So you don't have to just look at cartoons. This is at 3 in the morning in Bodega Harbor, but you can see all of the little guys crawling around. So they're usually, you find them at these really high densities, kind of flailing about. The amphithoids, let's see if the video is perfect. So here's this little head is right there. He made a little tube between 2 blades of grass, so they can sort of silk these grass together to make a little temporary tube. Or in this sad little dish, this piece of algae, they're trying to see this guy's kind of folding himself into the algae. So they also will sort of preferentially make tubes in these pieces of algae. And then this is isopod. It's harder to see, right? So they're pretty big. They hang out on the grass on the sides, and they look a lot like the seagrass. Okay, so stepping back, one reason we might be interested. One reason we might be interested in predation risk. Let's bring it down. That's okay. is to think about what that means for the different abundances of prey over time, right? So things are getting eaten, we might expect their abundance to go down. I think a lot of the time we sort of assume these kind of Lotka Volterra sort of cycles, right? Where predation risk goes up, abundance goes down, it recovers to a certain point and they cycle. But when we do this, the lag between when predation risk is high enough to impact abundance might be hard to predict. It might not be constant throughout time. It might change over different sites and across seasons. And the time scale at all this is happening, this could be days, this could be months, this could be years. And these things are difficult to sort of estimate without lots of sort of rigorous data. And then also if you have a prey community with diverse traits, if you figure out this relationship for one member of the prey community, it might not be the same for all of the other ones. So for my first chapter, I was interested in how seasonal predation risk varies by prey species, and then how this relates to prey abundance. And again, we're in seagrass. And so these are my methods. These are sort of the methods for all of my chapters. It's all kind of the same data set a little bit. And so I had 6 sites around Bodega Harbor and Tamaulipas Bay, out in Sonoma County, a couple hours west of us. And I went to these set transects for many years. So I set out these transects and then just kept coming back. And I did predation assays. So I took all of my prey species and I put a little bit of glue on their back and stuck them on a piece of string and then put them on little clear sticks out along my transect at low tide. And then I would come back 24 hours later, find all the sticks. If there was something missing, then I said that one was eaten. So my measure of predation risk is the proportion that is missing after 24 hours. So if all of the tethers have nothing on them, all of those things got eaten is my measure. And I put 20 out for each species. And then during this chapter, I also did prey surveys. So I went out along all these transects, and for 15 minutes, I was sort of like searched through the grass. And all of these guys are the biggest of the prey community. I chose them partially because they're easy to identify. So I can just count them in the field. And I got this measure of how many I found in 15 minutes as my abundance. Okay, so this is like the greatest video ever. There's a tether. There's an amphipod right there, but you can't see it, but the fish can. Oh. R.I.P. So one of them got it. Okay, so overall, the first couple of chapters, it was 16 deployments across the year. That was like every 2 to 6 weeks. Basically, every time I could go out there, I was out there. A lot. 7,140 crustaceans glued to string in our data set, and 13,372 were counted in the field. So now they're just lines. My N of 6. So here are the different species. This is the abundance, and the lighter colored lines are each of my sites, and then the darker line is the average across the year. And this is scale abundance because I'm interested in sort of how is the abundance going up and down? What is it doing through time? So all of these sites were scaled to the maximum abundance for that species at that site. And then this is the same species. That's my note to remember. If I call them caprellids, I'm talking about caprella. Amphithoids, I'm talking about amphithoid. And isopods, I'm talking about penidid. Because I forget that people don't use those names a lot. And so this is predation risk. So this is what proportion of the tethers was eaten at each time point. Again, the lighter color of the sites, the darker one is the average. So I looked at this data for like 3 years. I was like sitting on it. You can sort of visually see some patterns where some species are eaten more than others sort of consistently. There seems to be some sort of differences in the timing of abundance, but I just couldn't really quite figure out how to tell this whole story. So I think, classically when you have these time series data, what we'll do is we'll take like the time and the magnitude of the peak of predation, or abundance, right? And so if I look at the magnitude of the peak and the timing of the peak of predation, and then the timing of the peak of abundance, and I plot those against each other. Now on this y-axis, this is like the lag between when abundance was the highest versus when predation was the highest. You see sort of a general pattern, right, where caprellids are higher in abundance before they're peaking in predation. Amphithoids have some spread, and the isopods are peaking in abundance after predation, but this isn't like significant, and it feels like it was missing a lot of the nuance of all of the lines. So I went back and was like sitting and staring at it for hours. And then I heard about this thing called functional data analysis. So functional data analysis, the function in here is an equation, like a function describing a curve, not functional response or functional traits. And so this is a suite of tools that people use in lots of fields to look at the shape of an entire curve rather than just sort of summarize points around the curve. Okay, so here are all my predation risk curves pulled out now. Each of my rows is one of my species. Each column is a site. The dots are the observed points, so those are the actual measures I made. And then the line is a GAM that I fit to sort of smooth out and get an equation to describe that time series. So I'm going to show you a biplot next, and every dot on that plot is going to represent one of these curves. Okay, so here they are. So every dot here is now one of my predation risk curves, and they're colored by species. So isopods are this teal color, coprolids are that purple color, amphipods are yellow. And you can see it's sort of grouping out by species. And the cool thing about these functional PCAs is that we can figure out what the axes are actually telling us about variation in the curves. So this is the average of all of those curves right here. If you average all my predation risk patterns across all my species and all my sites, on average, one of the prey experienced this like unimodal peak around October. And this is the loadings from the first PC axis for my predation PC, which is explaining 72% of the variation across all those curves. And so what this is telling us is that if we're higher on PC1, we have a higher score on PC1, then it looks more like this average curve plus this loading curve. And if you're lower on PC1, it looks like the average plus the inverse of this loading curve. So we're like multiplying our score here times this plus this. And so to just visualize that, like a point over here that's really high, one of this amphipoid point, really high on PC1, it looks a lot like this PC1 loading, right? It looks like the average but shifted up. Over here, around 0, it looks a lot like the average predation risk curve. And then down here, it's like the average predation risk curve but flattened. So we get this axis that's describing this variation in our predation risk curves, and it's describing our variation in the magnitude. So higher PC1 scores is telling us there was higher peak in predation, lower PC1 scores, flatter peak in predation. Okay, so then I can do the same thing with our abundance curves. So now each of these boxes is abundance through time. If I plot them all on this plot, there's less clear clustering by species, but we look at our first PC axis, it's explaining over half variation, 52%. Our average scaled abundance through time is pretty flat. And this axis is describing what it looks like to be the timing of abundance. So you also notice the predation was peaking in October for all of them, and the flip in the timing here is happening around October. So if it's high on PC1, then it means that there was lower than average abundance before the peak in predation, before October, and then higher than average abundance after the peak in predation. If you're low on PC1 in abundance, then it's higher than average abundance before the peak in predation, lower than average abundance after. And so this sort of visualizes that. Isopod over there is peaking in abundance towards the end of this time series, right? So this is also spring to spring, so that's like late winter, early spring. The amphipoid around 0 has sort of a wiggly pattern, not sort of really explained by this. And then as we go further on the negative side, the coprolid is peaking in abundance earlier. Okay, so now I took this first axis from the predation PC1, describing our variation in the magnitude of predation, and plotted that against the first axis in the abundance PC1, describing our variation in the timing of abundance. And you see these cool species-specific patterns between temporal predation risk over time, and abundance over time. And they make sense with what we know about our prey traits too. So the isopods are clustering low on PC1 on the x-axis for predation, so they have pretty low predation overall. And they're high on this abundance axis, and so they're peaking in abundance after the peak in predation. So if we were going to make a hypothesis about how predation is influencing abundance, maybe we think that they're pretty good at avoiding getting eaten. So everything else in the prey community is getting eaten around October, and they've sort of shifted their life history to utilize this time with less competition. The amphipoids are consistently really positive on PC1 for predation. They always are getting eaten a lot no matter where they are, and they're really spread out over this abundance PC. So at different sites, they have different timings. And so remember, these are the guys that are seeking out algae to sort of make tubes, right? So an interpretation of this could be that they're always getting eaten a lot no matter where they are. So maybe they're more mobile and they're sort of using a spatial refuge more than a temporal one to avoid getting eaten too much, to tolerate this high predation across sites. And then the coprolids are around 0 on PC1, so they have this moderate peak in predation. They're always getting eaten a bit, but they're always negative on the abundance axis. So their abundance is shifted before that peak in predation. And then remember, these are the ones that are aggregating. And so maybe they've shifted their life history early to sort of reproduce quickly so that when the peak in predation comes, there's more of them and every individual one has less chance of getting. So yeah, so we find these cool species-specific patterns and they line up with what we know about the predator avoidance traits. Okay, so that's fun. So now if we look at all of the sites, I've just been hiding the sites from you. now if I put a key for the sites, we might notice that some sites, there's more variation on this graph than others. So if you look at this plus Nix Cove, even though all of these species are sort of within their own little species-specific pattern, they're pretty clustered. They're sort of converging there. Whereas other sites, the circle, Blake's Landing, they're very far apart. They have much more distinct patterns. So I was also interested in what drives site-level variation. So that's my second chapter. So I was interested in how do these species-specific seasonal predation patterns vary spatially, and then how do they, the species-specific patterns relate to overall community-level predation. And you can read about this in ecology as of yesterday. Okay.
Pause. I mean, that's good. Right?
Okay, so if we step back again, think about this theoretically. Another reason we might be measuring predation, right? We might be using predation assay too, is to think about sort of like total transfer up the food web, right? Maybe we don't necessarily care that much about species-specific differences, and we actually are interested in like an aggregate prey amount, right? In this scenario, each of these different colors is a prey species, and then the darker line is going to represent the community level average. And so I was interested in sort of what different patterns of species-specific predation risk might lead to different patterns of community level predation risk. Okay. And so in this situation, we have really high variation in the community level across the year. There's a time in the year where nothing's getting eaten and a time in the year where everything's getting eaten. And this sort of inherently has to come from low variation among all of the species. Alternatively, we could also see stable community level predation risk across the year. So there's always something that's moving up in the food web, right? And this comes from high variation among species. Either those species all are stable or they have different peaks and troughs that line up and make this temporal complementarity situation. Okay, so what might determine which of these scenarios happens? So now here are all the sites. Yay. So as I said, I did all this work in Tomales Bay and Bodega Harbor across all of these different sites. And these sites vary in lots of different ways. But what I'm most interested in for this chapter is habitat. So if we look at this site up here, Westside Park, up in Bodega Harbor, this is sort of what it looks like around the summer with the peak amount of eelgrass. So all those little green spots are little eelgrass plants. And so we have an increase throughout the summer and then a decrease in the winter. Some other sites that I went to, at Nix Cove, there's like 3 blades of grass here that come back every year. And so you have like a really small seasonal increase in seagrass, right? But it's pretty unvegetated, mostly a lot of the year. And then Blake's Landing, which is the closest site to Nix Cove, not very far away. In the summer, you have this beautiful meadow with lots of structure. And then even in the winter, when things die back, it's staying at a pretty high level. So there's always some habitat for things. And so I was interested in how might this habitat sort of either stabilize or make community level predation risk more variable. So here are all of those sites again. This is the same sort of data you've been seeing, but now it's in a different format. And so now each of our boxes is one of those sites. And these averages, these lines, are the average of all of those prey species. And so if you look at this, you might notice Nix Cove, that site that I showed you was brown, has very seasonal community level predation risk. And then Blake's Landing is pretty flat, and there's sort of this gradient along them. So these sites are ordered in terms of the amount of habitat structure, with lowest habitat up here, highest habitat there. And so I was interested in how does habitat relate to this variation. I took the standard deviation of each of these lines to get my measure of community level variation and predation risk, and then plotted that against the mean seagrass cover at each site. And we found this relationship where when there's not a lot of seagrass, community level predation risk is really seasonal. So there's a time of the year where nothing's eaten, a time of the year where everything's eaten. And when there's more seagrass, community level predation risk is sort of flatter. So there's always like 50% of some of my tethers are getting eaten. But I was interested in what species-specific patterns are underlying this, because remember, I just told you that all the species are different. And so how are they all getting eaten at the same time? And so I took my curves again. So here are the curves, and I did a different way of sort of putting these in multivariate space to look at differences. So I took this from the flowering literature. So this paper by Emily Austin, they were interested. So in the functional PCA, we get things that are describing actual variation, right? What I was interested in here is how different are the curves, not necessarily how they're varying. And so on these biplots, when points are closer to each other, they're more similar. And so the way we calculate that is we use these Kolmogorov-Smirnov distances. So it's a test you might have run if you were interested in testing for normality. It tells you how likely it is that one distribution was sampled from another. And so we can do these pairwise tests between all of our different distributions, and if they have a more dissimilar distribution, right? It's more likely they're sampled from different places. We'll have a higher test statistic. Then you can make a distance matrix and then plot sort of all of this into space. So this was from the paper that described this method. Points that are closer to each other are describing distributions that look more similar. Okay. So here are all of the lines that we're going to plot. So organized by site again, each of these different colored lines is one of my prey species at that site. It's predation risk through time with the communal average. You might notice I've added another species. This is Amphibialida. I included it here. It's like kind of complicated. It's like maybe an invasive species. It's only found at one site. I only tethered it into Mollusk Bay because it's maybe invasive. We didn't want to put it in Bodega Harbor. We were interested because it is a congener to Amphibialida certosa. They look very similar. It's smaller, but it has similar traits. And it was eaten at similar rates a lot of the time. So we've added another prey species, but don't worry too much about it. It's doing what the Amphibialidas are doing. Okay, and so again, these sites are ordered by low seagrass to high seagrass, and you might notice that at low seagrass, all these lines look very similar, right? Our species look similar, and at high seagrass, they become sort of more dissimilar. Okay, so now I'm going to show you a crazy graph. Okay, so this is every point here is a site, and these are all our KS distances sort of plotted in multivariate space. So points that are closer to each other have more similar curves, and each of these is a species at a site, right? So it's a species that has a similar predation risk curve to another species at that site. Okay, and then the ellipses are around each of the sites, and you might notice there is no clear site grouping. But if we look at just our two extremes, right, our Nix Cove with not a lot of habitat, Blake's Landing with lots of habitat, we see a pattern emerging. So this is Nix Cove, right? All of these points are pretty close to each other. And when we look at the graph, that is what we see, right? All of these species-specific predation risk patterns look pretty similar. And this is our site with low habitat. And then our site with high habitat, our species are the most dissimilar from each other. So the points are the furthest apart. And so all of those cool traits that I talked about and those unique ways of avoiding predation maybe are being more expressed at these places with more habitat. So I took the dispersion of each of these circles. So how different were all of the points at each site, and used that as my like among species variation in predation risk. So that's what this is. So higher dispersion, those points are more spread out. The species have more different patterns of predation risk. And if we plot that versus our community level predation risk, we can sort of see what might be driving that stable community level predation risk at our high habitat sites. So when there's not a lot of habitat, remember, over here, very seasonal community level predation risk, our low habitat site, all the species are also very similar to each other. They're lowest on our dispersion. When there's more habitat and our community level predation risk is flatter and more stable across the year, all those species are expressing more different patterns of predation risk. They have they're maybe able to use their traits more, right? They've evolved so nicely. So we see that high habitat cover maybe leads to this temporal complementarity among all these prey species. And so if we're interested in predation as this energy moving up the food web, it is more stable across the year. We have more habitat for different prey to do different things. And so finally, I was interested in what prey were sort of converging to drive this, what was causing that. So these are all the curves again. I looked at just, they all have this unimittal hump. So I just looked at the standard deviation of each of these lines as my measure of variation. And I looked at how seasonal, how variable are these curves over different levels of seagrass cover across all the sites. And for the species that have these more temporal refuges and also like more morphological traits to avoid predation, the crapelids and the isopods, as seagrass cover increased, they became less seasonal in their predation risk. It became flatter. But for the amphitheloids that are getting eaten a lot everywhere, that makes sense, right? They didn't change very much over different habitat cover. And so it seems like, habitat cover might also influence some prey species more than others and cause them to converge when there's not a lot of it. Okay, and so why might this be happening? Everyone always asks me about the predators. I love the amphipods, you can tell from the hack. But there are things eating them. And so thinking about different scenarios of habitat structure across the year, this line is representing how much habitat is at this site across different times of the year. And I'm thinking about what might be able to live there. So all these predators are also mesopredators, right? They're prey themselves. And then they also have a whole suite of unique traits themselves too, right? So some of them are also more worried about predation than others. So if there's not a lot of habitat, maybe we can only expect to see sort of the toughest, pokiest of the predators to be able to hang out there. So maybe we just see these green crabs. Then as in the spring, we get more seagrass growing. We start to see more sort of resident fish like pipefish that have some ways to avoid getting eaten. And in the summer, we have this full community with our migratory rockfish coming in, right? And so this might lead to sort of a moderate increase in predation risk across the year. At a site with not a lot of habitat, maybe across the whole year, the only things that can hang out there consistently without getting eaten themselves are these green crabs. In the summer, lots of big rockfish come in because they're not scared and they just eat everything up. Maybe that's what's leading to sort of our big peak that we see at Nick's Cove. Versus at Blake's Landing, where we have more consistent habitat across the whole year. Maybe the reason we see different species-specific patterns is because they can kind of consistently have a higher, like more diverse predator community that are able to eat different things at different times of the year. Okay, so I was interested in how, for my last chapter, how predator communities differ across this habitat gradient, and then how these predators relate to the predation risk size. So I did that 1 year of tethering. I went back and then I did it again. And so this time I did it 6 times across the year rather than 16. So here's the setup. We got our quadria to measure our percent covered. All these little sticks are the tethers. Wow. Famous. And then now I put up cameras. So ideally, I had 4 GoPros at a site. GoPros I found were very unreliable, so they broke and stuff. But ideally, there were 4 GoPros at a site. They were put out at low tide. At high tide, I could program them to turn on at a certain time. So I programmed them to turn on at the daytime high tide, following the low tide that I put them out. They would turn on, record for like 2 to 3 hours until battery died. We'd come back the next day and get them, and then watch and see what was on them. So before I talk about that, part of what was fun, we were trying to look at the predator community, right? But we also saw sort of fun little incidental things, which I enjoyed. So here's a little flatfish. He's hanging out in the seagrass bed, and in front of him is this big thing of ulva. And he's been sitting there for a while, and you might notice this little squiggle pops its head out. And so that is a little amphithoid. That's us. And so this amphithoid is hanging out in his tube. He's like, I have a great place to hang out. I'm hiding. But as soon as he pops his head out, the flounder sees him. So cool. So that was just fun. So we watched all these videos. By we, I really mean many wonderful undergrad. Which I have these like stock photos. We conference called it. Like so happy. They had us take these pictures. So many wonderful undergrads. We would watch all these videos. Every time a predator, so a crab, a fish, a bird, something came into the screen, we would identify it to the best of our ability. Again, mostly not me. I support. Identify to the best of our ability and record how big of a group it was in, and then how long it was in the frame for. And so then when I was looking at sort of these measures of predator community, our abundance measure is this rate, like how many surf perch or whatever do we see per minute is what I'm going to be looking at. Okay, so again, to remind you, we had all these hypotheses about how habitat structure at all these sites is going to change the predators, and that's going to change our predation risk. And then I look at it by site, and there's like no clear pattern. So each color here is one of our sites, and they're the ellipses. You might notice these 2 sites I've been talking about that have such different predation risk patterns overlap pretty totally in terms of their community composition. So we see very similar predator communities at these 2 places, which make, they're close to each other, so that makes sense. But when we color this by season and we look at seasonal patterns, then it becomes a little bit more clear that there is some structure happening here. So each of these different colors represents one of these months that we put the cameras out for, and then all the dots are the sites. What's cool about this too is that the colors are kind of hard to tell apart, but this like darker red is August, and this lighter red is July, and those were done 1 year apart from each other. So August was the first one, July was the last. So it does look like the community has sort of a consistent summer community. It shifts into this winter one and then shifts back. And so if we look at what are the species that are driving these differences, These are like the 20 species that have the biggest drivers in NMDS space. And there's actually there's a lot of things on here, but there are actually only really a couple of groups of species. So we see the rockfish. This makes sense, right, with what I said about these migratory things coming through. Sorry, I don't know why PowerPoint is like popping up on the screen. Okay, there we go. Thank you. No. Okay. If I go like this, yeah. Okay, so there's a rockfish. Wow. There we go. Okay, and then we have a lot of perch. So lots of different surf perch that are coming through. Also in the summer, pretty common, pretty fun. This is also what the videos look like. There's a lot going on. Okay, and then in the winter, the species that are sort of driving our winter assemblage are these ducks. So I didn't really think about ducks. Like was going out to all of these sites. I was going out to all of them, and I noticed that there were ducks in the winter. Like that's something I know, but I didn't really think about them as foraging in the eelgrass. But a common thing in these videos is that that was what would be happening. So this is a camera. We have these bufflehead coming in, and notice how much sediment they're stirring up. Shouldn't have done that. Look, so fun. So they're picking through the grass and they're causing a ruckus. There's a lot of stuff going on. And so, and sort of piecing these two things together, thinking about what can we say about these seasonal patterns. This summer, there's a lot of surf perch. There's still surf perch in the winter. The surf perch are residents, right? They are hanging out there year round in some capacity. And the ducks are coming in the winter. And so I looked at in videos, so that when the camera recorded, it split it into videos. So there's 11-minute chunks within the camera. So this is just in the winter months when the ducks have arrived. And in the videos where there was a bird present, so a bird present somewhere in 11 minutes, we saw less than half of a surf perch per minute, or per hour. But when there are no birds, you see many more surf perch, right? And so if you look at this across the whole year, this is the proportion of videos with diving birds across the year. You might notice they're sort of going up around October, December. And then this is the proportion of total time with surf perch in it. So as the birds arrive, there are fewer surf perch. There's even that little dip where there's fewer birds and more surf perch, which is kind of crazy. And then if you think about predation risks, these surf perch are kind of the equivalent of the amphipods that I chose to tether, right? They're at all of these sites. In some capacity, they are eating a lot of things. They're common across the year. So if you put this in a model, when there are fewer surf perch, there is also lower predation risk on all of the prey species. And the birds are arriving in October and they're leaving in March. And throughout all of this time, predation was always peaking in October every time I did this, and then would start to rise up again in March. So I never really understood why it would start to decrease in October. It seemed like a little bit late. I was expecting sort of a summer peak, So remember, predation peaks in October, but that's when the birds arrive. So I think maybe this is birds are coming in, they're scaring fish away, and we are seeing decreased predation because the fish are sort of scared by the birds. So, rather than habitat sort of filtering out different members of the community, maybe looking at the interactions between the members that are there and these complex interactions between habitat structure and all the different predators and all the prey that's available is what you have to do to sort of actually connect these predator patterns to fish. Okay, so conclusions. So for chapter 1, I was interested in how seasonal predation is varied by prey species, how abundance connects, and we saw that unique behavioral and morphological traits made these species-specific patterns of both predation risk and abundance. For chapter 2, I was interested in how the site-level characteristics changed predation risk, and when there was more habitat structure, there was more variation among all of the species and more stable predation risk at the community level. And then for chapter 3, how does variation in the predator community relate to predation risk patterns? And when it might be that sort of interactions between the predators and these behavioral things, maybe we'll explain more variation than sort of these snapshots of what's in the community at different places. And all of these insights were only possible because of high resolution temporal data and these tools to sort of look at entire curves rather than just summary statistics. And, as I think ecology is kind of moving that way, right? We have all these programs, we're collecting lots of long-term data. And as we sort of think about what to do with it, maybe some of these tools are what we do. And so hopefully I'll do some of that next. Yay. So in August, I'm starting a postdoc at UC Santa Barbara with Bob Miller and the coastal LTER and Holly Moeller to work on this project, looking at depth distribution of the different algal traits, but then also sort of to play with long-term data. So I'm excited. Yay. Okay, so I'm going to do like a boring thank you. So here's a slide with everyone's name. And then I'll do questions, and then I'm going to do a more personal thing. But if you don't want to see that, you can move. So I'm done for now. So thank you.
Yeah, so we'll let Claire take questions, but before we do, two quick things. One, it's her birthday today. Their birthday. Claire. And the other is that if there's any doubt about her ability to survive and thrive as a scientist, she taught 4 sections of Bio 1 today, and then gave her exit 7. So, she's already got it on all of us faculty. She's given it in one day. So Claire, go ahead and take questions, and I'll let you know if there's any on Zoom.
Okay, cool. Any questions?
Question about this one. So cool. I had a question about the functional data analysis. If you go not all the way back to that session, but just where you were comparing it to the bird abundance.
Oh,
things,
yes.
I saw there was this, like the PC1 loading. Yeah, looked suspiciously like the double peak that you were seeing with the bird presence too. Do you think that's a coincidence or?
Oh, so
I know it's different years.
Yeah,
so
it
I
is
know
different.
It's like literally.
Yeah, that's a good question. I hadn't been thinking about the years together. Because yeah, part of with this predation data, like you can see it is much less of a dramatic seasonal thing when you only do it 6 times, right? Like you lose a lot. But that's a good point. Yeah, I'm not sure if this was just like an anomaly. So one thing that I noticed with, I don't know what is happening. A lot of these, when you, when I first started tethering, I was doing it like every low tide basically. And then eventually I was like TIing and it was too much. So I was doing it once a month. And so earlier in the data, when it's more frequent, you see sort of like a tidal pattern too, where then there's a bigger high tide and a lower low tide. You have a little bit higher predation and then it goes down. So I kind of was like, it's probably some wiggle from when I was doing it early on, and it's like a tidal wiggle. But that is a good thought.
So now is the center of the graph the same month now that I look at it?
It's
No, okay, there,
shifted.
never mind. Yeah, okay, great, that is.
Yeah. Year time preference. Yes, Peter.
Hi, first of all, it was amazing. Oh my gosh, that was great. Did you ever have to worry about a consideration of like a spill or a pollution interference at all? Like, did that ever happen in any kind of data? And if so, what do you think it would do?
Like me polluting or like someone else polluting?
Eye in the beholder, sure. No, like a spill, like something coming like an inlet from a harbor or like a rain, like significant rain event, an algal bloom, something like that.
Yeah. So in this first chapter, there actually are only 5 sites in here too. Because one of the sites is that amphitheoid, the other one, it's only found at one place. It's found in the back of Smallest Bay. And like basically the only amphipod that's there is that one. And it's super yucky there. It's like if it rains, like Emma and I went out and lab tech and like we had to cancel a bunch of tetherings because we were like getting a rash from just sitting in the water. It was really gross. It's very safe. I didn't say that. So I mean, yeah, there were a couple there was like an oil spill at Split Point once, but I didn't necessarily see impacts. But yeah, that was the only thing. But I'm sure there's lots of gross stuff in Mars. Yeah, Rick, I guess.
In the first chapter, you told us that the prey might gain some advantage, that predation risk for any individual would be lower if they were more abundant. And it seems like for that kind of argument to work, there has to be some reason that predators don't just move in and eat more of those prey? What limited sort of predator recruitment to those kinds of places?
That's a good question. I think my instinct is like from a predator standpoint, you're not ever really gonna have, there's always gonna be something available for you to eat. Like I think the predators are maybe moving less in terms of what's available, just because these guys are like I could go out and count, 600 caprellids, but like 600 caprellids at a bunch of different sites, right? And it's like 600 versus 400 versus 500. So I think, and then you still are counting like, 15 isopods in like 15 minutes. So my sense is sort of like as a predator, other things are limiting your movement more than where the prey are. But that is a good.
Like what?
So they're on a really strict temperature gradient, right? I think if you are a mesopredator, your own predation risk is probably important, right? Yeah, I mean, I think, I don't know the mind of a fish, I guess. Like, I feel like, well, the flatfish, if they're hunting for amphithoids, like maybe you're looking out for ova instead. Maybe the caprellids, a lot of times, I think they're eaten by like these schools that are sort of going through and browsing too. So, and a lot of the videos will have in like these smelty sorts of things go through and pick things off of the leaves. So I think there's like a lot of nuance in terms of like what is eating what and what is sort of driving what they're eating at different times of the year. That's just kind of like a cagey answer, but I don't know. Yeah.
First, I'm going to maybe comment on Rick's question and then ask what I was going to ask. But yeah, for that to work, that school effect. So it's usually like a confusion effect that makes an individual sort of hard to pick out. Or if something isn't that tasty, then that can also be protective in a group where a fish eats one and goes, I was in the mood for peanuts, not popcorn.
Yeah.
So anyway, so those are the comments there. And then, back when you, by the way, great job. I should have started. I mean, that's just such a confusing data set at first blush, and then what you got out of it is spectacularly informative. Way back when you proposed this, I was wondering whether there would be a seasonal fish recruitment that would affect the amount of predation in different months. And so I don't know whether you saw anything in the size structure of the fish when it was getting towards the peak, or maybe you could just comment on that.
Yeah, I think I had like all of these hopes and dreams about the fish data, and then it's like really hard to get things out of them. So like I showed you sort of like, you might notice on this NMDS, like some of these months have fewer points than others, right? So I had a hard drive that broke that we lost a bunch of videos for some of the months. And then sometimes the cameras would sort of fall over and ova would get caught in them. So, and then trying to figure out the scale. So like we have some data on, we tried to record if something was had like a juvenile patterning or seemed like a juvenile versus an adult. And I was looking at it and it's basically like everything was recorded, we recorded them as juveniles across most of the year. So I think, yeah, I'm sure that there is like recruitment coming into different places and stuff. I think when I looked at, the only pattern I could find with abundance was if I look at just the total number of fish, the rate of fish across of all of the things that I'm seeing, all the predators at a site over time for each site, then look at the variation. At Nick's Cove, the site with really variable predation risk also has the most variable predator abundance. And then there's like this correlation where as the predation risk becomes less variable across all of my prey species, the abundance of all the fish becomes less variable across the year. But I think it's a good, I'm sure that there are different recruitments happening. I mean, rockfish, I think, are like only coming in sort of when they're young. Yeah, I don't know if that answered your question.
No, that's data are noisy, but I'm glad that you looked at it.
Yeah.
Really great talk. I just had like a natural history question, which seems like you might be the right person to answer it. You talked about how seagrass is like a flowering plant, and then you got all these little bug-like things crawling around all over them. Like, how is seagrass, like, pollinated? Do they have a relationship? Is like sort of that?
Yeah, that's like a whole can of worms. So it's like water pollinated in some ways, right? So there is this paper that's like the isopods are the pollinators and they're the bees and you go and use it. But I feel like people don't necessarily agree. Yeah, it's interesting. There's lots of like cool plant herbivore connections between the seagrass and the amphipods and isopods and stuff, but doesn't totally, the whole analogy doesn't hold on. Yeah, some of them, that's the other thing is like different members of this prey community are eating the grass versus they're eating the algae. So the grass can also respond by making chemicals to make it less palatable for different, but yeah, there's a whole thing. Yeah.
Yeah, that actually made me think of a question. So yeah, some of the species eat like the epiphytes and stuff off of the plant, whereas some eat the plant itself.
Yeah.
Does that have any difference on like, I guess it's like strategy for avoiding predators and stuff?
Yeah, I feel like
Let's do what?
yeah, so one thought is it maybe like ties into the abundance pattern of the isopod. So like with all of those abundance predation patterns in the first chapter, like I can't say that predation is causing those things to happen, right? There's lots of drivers. Like isopods are eating the grass. And in the winter, when they're more common, is after the peak of predation for everybody else. It's also when the grass is sort of more palatable because it's breaking down. So I think there's lots of things that all interlace. Yeah, the amphithoids like to eat ulva, so if they're moving around to hide, they also might be moving around to eat things. The predators are kind of just like catching things. But they're also all pretty, like, most of these guys are pretty generalists too. They can eat lots of different.
Cool. Well, let's thank Claire. And feel free to duck out if you don't want to see the acknowledgements. Otherwise, I'll let you go ahead.
Okay, I have some fun pictures. I have lots of people to thank. Also, I was making all of these slides. I was like, everyone is always like, oh, you can't fit all the pictures. It is really hard to fit a lot of pictures on these slides. So I tried. It won't be that long. It'll be like a couple minutes. But I do feel like I have to formally thank people. So first, all of the undergrads and lab techs who helped me with all of this. So lots of people. There was like lots of time in the field. Lots of different people came out. From Laura, sort of helping develop the tethers, to SRJC interns like Cassidy coming out, Victoria. Yeah. And we had a lot of fun in the field. So here is some fun times in the field. So thanks to everybody for being silly with me. But this was the first round of tethering that gets excluded from the data set because you might notice everyone's falling over. All these people drove from Davis to come help me, and then we like broke the transect tape, like holding on to it. And Caroline came out, and we were like recording videos in the rain. We had this grand plan to make all these videos, and then it was pouring rain. You can't see anything in the videos. Maddie came out, and this was like early when I didn't really know what I was doing. We both lived in Davis, and we like drove out in the night to like get one thing, and then we drove back in the night. And there's like big waves. We've gone out in the night. We've seen cows on beaches. We've had lots of fun. Everyone has put up with me loving a gimmick. So we've had multiple Halloweens in the mud. Emma and I dressed up like Caprellas and amphipods when we went out and did field work. Serena and I dressed up like superheroes when we went out and did field work. When I finished tethering the first time, Emma and I got 3 different cakes, one for every site. And then it was like pouring rain and we ate them in the car. And then we finished tethering the second time, we got another bigger cake. One time Emma and I got really excited like halfway through the first year, and we decorated these waders with our names on them, and then we put them on and realized we got child-sized rings. I appreciate that all these people, especially Emma, like really Emma Dean was out a lot, but everyone has had a very good sense of humor. Like I've taken lots of people out and made them do lots of silly things, and also just like walk them through the mud, and everyone has very much enjoyed it. I've enjoyed it. Lots of people have come out and held things. So. Holding fishes. This is Katie Dubois, like taking me out in the first summer before grad school, and she was like, "Close your eyes," and then gave me this giant sea slug in my hand. It was crazy, this big aplysia. So yeah, I appreciate all of the people who have helped me, both sort of through employment and also through friendship. I've sent lots of emails being like, please, please, please come help each other, please, please, please. And everybody's come and done it and worn silly hats. So thank you all. I have to thank Jay and the Stack Lab. I don't have any like pictures of just us. So, but I do have, I do have this video which I've been saving. So I don't know if the sound is gonna work.
Oh, that'd be too bad.
Well, it's this video, there's Thunderstruck playing of us walking through the mud. Because I don't know, this when there's you guys need help with these like high schooler drone people, and then there was this man who took all these videos and put them on his Instagram story. You can see it was like 10,000 likes. There are all these videos of us with filters. So I've been saving that literally for like 4 years. Thank you to Jay, who did help me lots of field work. Like so many times, I was like, I'm gonna go out in the middle of the mud. I don't have anyone to go with me. And he would drive from Davis, come out in the mud with me, and then drive back home the next day and like go teach Bio 1 or whatever. I have gone to his office many times and been like, I have just one question at like 4:30, and then I have like 7 questions. He's very patient. He's always been available when I've needed him. I feel like when I started grad school, I like couldn't think in a straight line. All of my thoughts were sort of all over the place, interlacing like all of the circles on my graph. I think working with Jay, I have become much better at sort of thinking clearly and thinking. So I appreciate it. And I appreciate the Stack Lab. So look at how fun we are. Here's all of us at a conference. This is like one of our first days of field work, our first summer. Carolina, special shout out, been there from the start. Jay likes his Christmas decoration. So yes, Stack Lab has been a wonderful source of support. I don't live in Davis and haven't really for most of grad school. I've been commuting from Lamell, but I always have had a desk. Well, most of the time I've had a desk. And everyone's been very friendly. They've all come out and helped me with field work and stuff, even though they don't live out there. I appreciate it. It's been good fun. So thank you to my committee, Jay, Eric, and Marcel. And then also I have to thank people who sort of got me into science earlier on. So John, JJ, and then Julia, who's at North Dakota State. So I, have always been fond of little bugs and stuff, and all of these people have sort of shepherded me towards them, which I appreciate. I don't really know where I would be if John and JJ had not introduced me to the amphipods like 10 years ago, and encouraged me to keep asking questions about things that people don't care that much about usually. Thank you to all of these large institutional groups of people. This is our cohort. I think this is like the best picture ever. No one's used this in an exit seminar yet, but we're so cute. We all started during COVID, and we like saw each other like 6 times in person in parks, and it was always a really big deal. So we did a photo shoot. I worked a lot with the Santa Rosa Junior College internship program for a couple of years over the summers. It's been really great. I learned a lot from it, and like we matched interns from Santa Rosa Junior College with people at the Marine Lab, and they've all been wonderful, and that's been great. Pop Bio, the Bodega Bay Bat Stars, lost every game. VML, which I'll do another talk where I thank them more profusely when I go out there, but I've been based out at the Marine Lab for most of my PhD, basically, and like, yeah, I very much appreciated all of the people there. Again, always willing to help me. When you're sitting, especially like if I'm not TAing and walking to Davis as much, especially Eric Sanford has sort of stepped in and given me a place in his lab when I needed one. Anya Brown has helped a lot. So I appreciate that for giving me more of a community out there. Okay, then all of my friends, yay. So I have so many pictures. I'll just show them all to you. So here's one. I made 2 slides of pictures, but I appreciate all of my friends, both grad school friends, non-grad school friends, everybody who still loves me, even though I just talk about amphipods. Yeah, some more friends. It's hard to include everybody. But I don't know, everybody and more. I have to thank my family. So all of these pictures are just my sister taking selfies. It's the only time we have family pictures, apparently. So my aunts, my siblings, both my sisters, my cousin, all have been very supportive of me. Again, don't really understand what I do. Very nice to me, which I appreciate. I have to thank my mom. She passed away last summer, but very supportive of me the entire time. So I don't have a lot of pictures with her because she didn't like taking pictures that much. But here's when she graduated from law school. And I do have pictures that she took of me. So when I was a little child, when I was 7, all of the cicadas came out. And I loved the cicadas. And my mom was not like an outdoorsy bug person. And instead of being like, oh my God, like my daughter is kissing a bug, she like took a picture of it, and then kept it, you know, like she like had this sort of at our house. She would show it to me all the time. She did not understand why I was so excited about like these little creepy crawly things and being outside, but was like very supportive from a young age. And then my first year of grad school, I didn't go home for a whole year. It was COVID. It was like the longest I'd ever been away from home. And she would send me, she like walked around the park by her house and would like find all of the best places to see the cicadas, and was like sending me videos of the cicadas, and was like on cicada alert. So then when I got to come home, she took pictures of me with the cicadas. So she isn't here, but I appreciate her for supporting me to do what I want to do. And so with that, I have to thank the bugs. So yeah, so you know, you have to thank the natural systems. I've been very lucky to work in these beautiful places, see lots of really cool things. Beach Hopper, RIP the Beach Hopper chapter. This was on my birthday 3 years ago, actually. An octopus took my data. Yeah, so